Hello, I am Paolo Grigolini, professor of physics at the Center of Nonlinear Science of the University of North Texas. I am directing the research work of 6 PhD students to reveal the role of criticality in biological and sociological systems. This article proves that a realistic model of interacting neurons may fit the complexity matching condition. Complexity matching. What's all about? This slide illustrates a recent experiment done at Duke University. A few electrodes are implanted in the brain of rat A, and with a cable are connected to a few electrodes implanted in the brain of rat B. The information on the dynamics of brain A is transmitted to brain B. The motion of the whiskers of rat B is proven to synchronize with the motion of the whiskers of rat A. In this article, we reproduce the result of this experiment by means of the dynamics of two neural networks representing the brains of rat A and B, respectively, and we prove that this is a manifestation of complexity matching. Complexity matching is the phenomenon of synchronization between two networks A and B when both are at criticality. The brain dynamics are characterized by events of intensity S and by inter-event times denoted by the Greek letter tau. Criticality is expected to correspond to either the scale-free distribution of the intensity S, or of inter-event times tau, or of both. In the current literature on neural networks in the brain, there is a general agreement that these systems work at criticality, but there is not yet consensus on which is the proper signature of criticality. Our work proves that in neural networks, the proper signature of criticality is the scale-free distribution of inter-event times, a property that we call temporal complexity. Increasing the concentration of inhibitory links makes larger the strength K necessary for the system to reach the criticality condition. On the other hand, if the number of excitatory links is excessively large, the system is insensitive to the environment. Thus, the inhibitory links have the beneficial effect of making the system sensitive again. The figure of this slide depicts the cross-correlation between the network B and the driving network A. The two networks are identical, and the concentration of inhibitory links is identical. The value of the cross-correlation, denoted by the red line, in the absence of inhibitory links, would be negligible. It becomes maximal when the concentration of inhibitory links is large enough as to bring back both networks to criticality. We believe that this figure affords important suggestions for the future research work, which is expected to describe the self-organization of neural networks toward criticality. The new theory should require that concentration of inhibitory links is not fixed, but let free to change so as to realize the maximal sensitivity of the system.